So I just start? Okay,
Yep.
sorry. Um, the similarities and insights between the epidermal differential genes in the Gallus genome and the Myriaclidia genome and how they compare. The unique development of the turtle shell contrasts with other vertebrates' protective structures. Research on the red junglefowl chickens and turtles suggests that evolutionary changes in the integumentary structures are linked to adaptations in the EDC-related genes. Turtles have expanded EDC genes, contributing to the shell, while chickens benefit from calcium bindings for health and egg composition, highlighting crucial molecular adaptations and epidermal differentiation for both species evolution.
The complex that holds the genes to form a green sea turtle shell is the same complex for human skin in the epidermal layer of chickens. This complex is the epidermal differentiation complex, and we observed it on human chromosomes. It was clear, however, that all the resulting phenotypes were different. Looking further, it was discovered that the turtle genome had a unique version of the EDC. Therefore, the question arose, if the EDC resulted in the turtle's shell, then how is this related to the EDC in chickens? Studies were read about the similarities between chicken and turtle genomes having 6 chromosomes almost equivalent to each other by Masuda. This was supported by other stories that were included in the remainder of our project, and the objective of our project in total was to examine the EDC of the turtle in relation to the chicken genome, since the end phenotypes are different.
So to accomplish this objective, we used the list of genomes provided by Michke that in the study from 1996, this list of genes specifically, and their counterparts or equivalents, because the list was in the human form of the gene, and so their equivalents were found for the chicken form of the gene and for the turtle form of the gene. But the chicken form of the gene, as you can see in the second column of the table, was used to run a BLAST-P search against the Myriaclidia genome family. Each gene was put in and a BLAST search was run, and a similarity score and the query coverage was recorded. There were, however, several genes, as you can see down at the bottom here, that had no equivalent for the chicken and turtle genome, or we also found one gene that was completely not similar. They just were completely different in the chicken and turtle. So what we found was that the calcium binding genes, as you can see as the first and second, and the first, fourth, and fifth row, those were the most similar with the best query coverage. The structural proteins and things like that were not similar, which would make sense that the structural proteins are very different, as this loricrin protein that seemed to be very critical in the epidermal differentiation complex, which comprises over 70% of the cornified envelope of the turtle, which is the cornified envelope is like the outside cornification of the shell, is very different when compared to the chicken. There was no similarity found, and that makes sense because there is no shell for the chicken. We found distance trees for the BLAST results, and we will put them on our poster because we think it's very interesting. To scroll through them quickly, we found that the chicken genome was always most similar to the ocean turtles of the Myriaclidia genome instead of anything else, which we thought was very interesting. And we have also included the alignment queries to show the percent similarities and the actual query coverage.
And for our discussion, we compared it to our results relating to how we found several proteins apart from the original epidermal differentiation complex that we originally provided. And as we mentioned in the results, those did not exist in the turtle Myriaclidia genome or Gallus genome, or no equivalent. And the figures and table will also show the similarities between the chicken and the turtle genomes, specifically also within the S100A10 and the S100A9 gene, focusing on their relationship and their functions. And these findings, as we also mentioned in the results, are calcium binding proteins of the EDC in genomes of both organisms, meaning that most of their proteins would be somewhat similar looking at the trees. And it's interesting that the calcium binding proteins were so similar between the two species. A future study would investigate this further to perhaps find out why. So then we also find these questions at the end in our conclusion. How do these calcium binding proteins function in relations to their surroundings? Does this mean that the calcium binding pathway is similar to the Gallus gallus and the Americalidia clade? So that's just our project and our study.
Do we know